So today we're going to talk about network visualization. So we saw how lists of genes or proteins that are identified as differentially expressed when comparing disease cells versus normal, or perturbed cells versus control, or if you have a list of genes identified as having mutations that cause a specific phenotype, or list of targets of genes for transcription factors as determined by ChIP-seq. They all can be analyzed for functional enrichment, and we demonstrated this in the previous lecture using enrichment analysis tools such as Enricher. However, we can also, as we've seen with Genes2Fans, connect lists of genes or proteins to form subnetworks given a large background network of either protein-protein interaction networks or functional association networks. So those subnetworks that connect those seed lists of genes can reveal relationships between the genes or molecular complexes that can directly be interpreted with specific functions. In addition, as we've seen with Genes2Fans, these additional genes or proteins that get pulled into those subnetworks to make the connections between the seed nodes are interesting and potentially can become great predictions that can be validated experimentally. So in this lecture, we will learn more about how to build those types of subnetworks and how to visualize them using a variety of network visualization and network analysis tools. So our lab was one of the first to develop a web-based tool that takes in a list of mammalian genes or proteins and connect them using known protein-protein interactions. So for creating this tool called Genes2Networks, we first merged from the literature several databases, online databases and publications that described many protein-protein interactions that were collected either experimentally or from literature curation. We then created a simple filter to clean up the database by removing papers that reported many protein-protein interactions. And this took care of the high throughput types of methods that were considered to be less reliable. So the tool takes as input a list of human or mouse genes and then builds a subnetwork using known protein-protein interactions, implementing the shortest path algorithm that we went over before. The example that we look at here is when entering the 4 Noonan syndrome causing genes KRAS, RAF, PTPN11, and SOS1, and we can look at this example online. So this is the Genes2Networks website. We're gonna click on Add List, on the Add List link, and type in the 4 genes that are the Noonan syndrome causing genes that we want to investigate. Then we'll click on Upload. So Genes2Networks also have some properties that you can adjust, such as the path length, the significant cutoff, which will adjust the colors of the intermediates, and the various databases that you can choose from. I'm going to unselect the last 2 databases, and this is the resultant subnetwork that I received when entering those 4 genes. One of the things that I would like you to note is that the program also outputs the significance of those intermediate proteins, and the first most significant protein, SHOC2, has 4 links in the background network that has 11,000 links, and 2 links to the seed nodes, to the 4 seed nodes, while they're in the subnetwork there are 22 links, and that gives it a very high z-score. So let's go back now to the presentation. So this analysis worked well for this particular disease, since mutations in this disease are within the same pathway. Noonan syndrome and Noonan-like syndromes are caused by a mild upregulation of the MAP kinase pathway. SHOC2 was predicted to potentially be involved because it had 4 known interactions, 2 of them with the Noonan genes that were known at that time. And when we set the path lengths to 3, all the 4 links for SHOC2 were present in the subnetwork. Using experiments that introduced a mutation that was detected in 5% of the patients with Noonan syndrome, but without an explanation of the genes that is mutated in the disease, show that there is a mutation in SHOC2 that could potentially be a functional mutation that caused the disease. Bruce Gelb and his colleague then showed that the mutation caused a missorting of the gene product, and that causes a mild upregulation of the MAP kinase pathway. Missorting means that the protein was going to the wrong part of the cell. The patients with this mutation have a Noonan-like syndrome that is associated with a typical antigen hair. Identifying disease-causing gene is important for potentially fixing those mutations, but currently they are used in prenatal screening. This company called Genes2Networks added SHOC2 to their test. Our best implementation of the idea behind Genes2Networks is a part of the software expression to kinases, which we will focus on also in the next lecture when we talk about the idea behind expression to kinases. Let's try to work with expression to kinases to build a subnetwork for the same genes that we entered for testing enricher. So first we will go to the expression to kinases website. We can download the entire program. If you're interested in individual components, you can download the Java jar files by download individual components link. In our case, we'll use the main installer and install the expression to kinase on our desktop. So now we downloaded the jar file, the install jar file. We need to have Java in order to extract and execute this jar file. So we install Java. Now we can execute the installer. You can also install the source code of expression to kinase if you are a Java programmer, or if you're interested in modifying the program or learning about the code. And now we're done. So now we have expression to kinases installed with a shortcut on our desktop. So I need a list of genes. Just to demo the protein-protein interaction functionality in expression to kinase. So I'm just going to go back to the enricher website, and I'm going to click on this regular example demo. And I'm going to copy all the genes from that regular example. So I have a list of genes in my buffer. Then I can close this. So now we can fire up expression to kinases, and we can select the protein network tab. And in the protein network tab, you can cut and paste the list of genes from enricher. We also want to specify an output folder. So we're going to call it demo PPI. So now there is an output folder for where to put the networks. We also want to check Cytoscape and Pagic as optional networks that we want for our output. Here we can also specify the databases that we would like to include, and here there are many more databases compared to what you've seen for the initial application of Genes2Networks. We can also modify the path length. So we can have 1 would mean just find connections between the input list of genes. And this is very good when you have very long list of genes. 2 is probably the most commonly used feature, where you get only the first neighbors that can connect at least 2 genes in my input list. And 3 is when you have very short lists and you want to find connections between very few genes. You can pull in many interactions. So if I click Expand Network, the Expand Network button, now it generates a network that connects those input gene lists. And now I'm not going to view the results within this tool. I'm going to go and open the file in the folder that was generated. And I'm going to click on the YED network. So I will have to install the YED program, and then it will identify the demo PPI graph ML file, associated file, and then I can just double click on this file. It will fire up the YED application, and it would display the network in this format. So the first thing I would like to do once I started YED, I want to change the layout to organic, and I'm going to just use the defaults. And you can see that the network is displayed where the seed nodes, are the red nodes, are connected through those green nodes, which are additional proteins that make connections between the seed nodes. So you can explore yourself many of the options and features of YED. It's a very powerful network visualization program. It has very good tutorials and help, and there are some YouTube movies that you can watch showing you exactly how to create more of those networks. At least now you have a pathway starting from lists of genes to find their connections using protein interaction networks or functional association networks to generate those images that then you can customize and adjust in software tools such as YED. Another very important and powerful network visualization tool that is probably the most popular tool in systems biology is called Cytoscape. One of the most important features of Cytoscape is that it has a plug-in architecture that allows many developers from across the world to add to the system various features that can be used to, for example, identify modules in networks, perform enrichment analysis, and visualize enrichment analysis on the Go network. Cytoscape also has a lot of tutorials and help that you can utilize for your studies, and it's highly recommended. One of the challenges for novice users who wants to build network is how to automatically draw a network from your data. For this course, we've developed a tool that can help you with this initial step of converting a network stored in a text format or in an Excel spreadsheet into those network diagrams that can be loaded in tools such as Cytoscape, YED, or Pagic. The name of the tools that we developed is called Text to Graph. So the tool has two tabs: network from text files and networks from gene lists using protein-protein interaction databases, which is a very similar implementation of Genes2Networks as you've seen in the previous slides. Text to Graph is implemented in Python. It takes two text files as input. One describes interactions or links, and the other describes properties of the nodes. You can execute the tool from a command line, but there is also a web application. This web application is extending the D3 library for network visualization. D3 is a JavaScript library developed by Mike Bostock, who I'm going to discuss a little bit more in more details in the next lecture. The Python script generates Cytoscape, YED, and SVG images from those two text files that describe properties about links and nodes. I highly recommend you to test it yourself. There is also a PowerPoint presentation tutorial that Chris Stan, who developed this tool, created for you to get you going. One more tool that does a very similar task connecting list of genes or proteins using background network of either protein-protein interactions from literature or functional association interaction is string. This popular tool has a very powerful web interface, and this is one of many tools that can be used to build subnetworks from lists of genes. In the next lecture, we will discuss an alternative way to visualize networks that is not the ball and stick diagram that we reviewed here. One of the challenges with ball and stick diagrams is that once you have many nodes and many links, as you've seen in the example of the YED network visualization, it's very hard to obtain much information from those complicated network diagrams. So we will show you how there could be some alternatives, how to visualize networks on a canvas.
Your courage, your cheerfulness, your resolution will bring us victory.